Okay, it's really great to be able to participate in this meeting. The London Metallomics facility is quite a phenomenon, and I'm just so pleased to be celebrating it today. That said, actually, my lab does not do any metallomics yet, but what I'm going to tell you about is our studies of the epidermis and how we use different techniques to explore the properties of the different cells within the epidermis, in particular the role of the Notch signaling pathway. So to start off, this is a histological section through healthy human skin, and it shows that the skin is made up of two layers: the epidermis, which is a multi-layered epithelium, and that is separated from the underlying connective tissue, or the dermis, by a basement membrane. Now, the epidermis is one of the classic tissues where it was appreciated many, many years ago that there must be a population of stem cells. That is, cells which make more cells like themselves, but also produce daughter cells that will undergo further differentiation. And the reason why that is obvious is if you look at the outermost layers of the epidermis, these cells don't have a nucleus. And so when they're shed from the surface of the skin, they must be replaced through a process that involves division of less mature cells. We know from many years of work that most cell proliferation or division takes place in the basal layer of cells attached to the basement membrane. And then as cells leave this layer, they undergo a process which we call terminal differentiation and move towards the surface of the skin. This journey time varies between body sites, but it's typically about a month. So we're interested in the stem cell compartment of the epidermis. We're interested in differentiation. But another feature that I want to point out that people sometimes don't notice is that the boundary between the epidermis and the dermis isn't flat. You can see it undulates. So at certain points, like down here, the distance from the basal layer to the surface of the epidermis is longer than in regions here. Now, my interest in the epidermis starts really from when I was a postdoc at MIT. And over the years, many of us have been involved in the exciting process of trying to find out what the markers of the different layers of the epidermis are, and also what the genes are that regulate their behavior. So what we're looking at schematically here is the basement membrane in gray, the basal layer of the epidermis with the stem cells, and then this process of terminal differentiation, where we know a lot of the changes in gene expression which characterize the process. Now, my postdoc was the late Howard Green, shown here, and he, back in the 1970s, showed that you could isolate cells from human epidermis and grow them in a culture dish at clonal density. And as the colonies expanded, they merged with one another and made multi-layered sheets of cells. Now, although Howard never practiced medicine, he did train as a doctor in Canada. And I think probably because of that, he was well aware right from the get-go of the potential clinical importance of his discovery. And it's quite remarkable that only 6 years after the original in vitro publication, he was able to show that you could graft sheets of cultured epidermis onto a burn injury and it would essentially permanently recover the damage. So this is a very early example of how you can expand stem cells in culture and use them as a beneficial treatment in patients. Now, the word that I want to highlight here is the word colonies, because the fact that the cells could be grown at clonal density really gave us a handle on what might be a good marker that we could use for stem cells if we're looking at cells in culture. And so my lab and many other labs count the number of colonies that are formed by epidermal cells when we plate them in culture. This is actually some dishes where we were screening different batches of culture medium to look for which supported the growth of the cells best. And we've seeded the cells, grown them for 14 days, and then fixed and stained them. So these individual plaques that you can see here are individual clones of cells. Now, this is, of course, an imperfect assay because it's purely in vitro. But it's quite interesting that in the wonderful world of dermatology, there are now many examples where you can actually see this sort of clonal organization in living skin. And this is an example of a patient who is heterozygous for a disease-causing mutation, and the heterozygous regions of the epidermis look completely healthy and normal. But where individual cells have acquired a second disease-causing mutation, there you can see these little plaques emerging. And we know that these correspond to clones of cells founded by a stem cell. So with this, by way of our knowledge of the epidermis, my lab set out many years ago to try to find markers of the stem cell compartment. And the approach that we took initially was a candidate approach. And the first marker that we went looking for was expression of the receptors which attach the basal layer of the epidermis to the underlying basement membrane. And we showed using flow cytometry and ECM adhesion assays that we could enrich for these colony-forming cells by selecting the cells with the highest levels of the beta-1 integrins. And we also found that the cells that express high integrin levels are not randomly scattered throughout the basal layer of the epidermis, but they're clustered. And that's a phenomenon that has been of considerable interest to us over the years. Another finding, which is really illustrated quite well in this schematic from almost 20 years ago, is that integrins are much more than simply Velcro that attach cells to a sticky surface. We know a great deal about their role in signaling, and we know that they control many core processes, including proliferation, motility, cell survival. And in work that we published long ago, we showed that in the epidermis, the beta-1 integrins signal through the classic MAP kinase cascade, and that signaling pathway controls exit from the stem cell compartment. So we've found our first marker of stem cells, and we found that it's not only important for cell adhesion, but also regulates differentiation. The second marker that I want to highlight from a historical point of view is this protein called delta-like 1. This is a ligand for Notch receptors, which I'll tell you a bit more about later. And it's quite interesting that the reason that we found this was because at the time we were working next door to some Drosophila geneticists, and they were really excited when they realized that what seemed like classic Drosophila genes actually had mammalian homologs. And so we took advantage of this to go looking for the human homologue of Drosophila delta-like 1. And we were able to show by northern blotting and in situ hybridization that we could detect delta-like 1 expression in the basal layer of the epidermis. And our functional studies have, since then to the present date, largely relied on delta overexpression. So the Notch pathway is one that is important in very many cell types. And essentially we have Notch receptors, which you can see on the surface of one cell, interacting with Notch ligands, like delta-like 1, on the neighboring cell. And we know a lot about the pathways downstream of Notch and its ligands. And we know that it tightly regulates transcription. So in our studies that we published some time ago, we found the interesting result that if you overexpress delta in stem cells in culture, it stimulates neighboring cells to differentiate. So here's a cluster of cells shown schematically with high levels of full-length delta-like 1, and they are instructing the cells with wild-type delta-like 1 to differentiate. By manipulating the cytoplasmic domain of delta-like 1, we were able to show that we could interfere with cell-cell adhesion uh, uh, independently of interfering with the differentiation signal. So delta light 1, like the beta 1 integrins, is a marker of the stem cell compartment, but it acts in a different way, telling neighboring cells to differentiate and ensuring that the stem cells in the basal layer of the epidermis are clustered. So conclusions from these studies in culture, and from many others that I haven't described, are that there are multiple populations of keratinocytes in the basal layer of the epidermis. The stem cells are more adhesive to the extracellular matrix and to their neighboring cells than other basal cells. And we know that ERK-MAP kinase signaling is required to maintain cells in the stem cell compartment. And as I've just told you, the communication between stem cells and their neighbors is such that they send signals to tell the neighboring cells to differentiate and leave the basal layer. So that's by way of an introduction to the stem cell compartment in the epidermis. More recently, we've turned our attention to what we call the committed cells. So these are cells which are still in the basal layer of the epidermis, but they're no longer stem cells. They haven't yet left the basal layer or begun to express differentiation markers. And we've been really interested in what it is that makes a committed cell distinct from a stem cell. Our approach to looking at this was to go back to another old Howard Green experiment, which he showed, in which he showed that if you are growing cultured human keratinocytes in a dish and disaggregate them, make a single cell suspension, and then hold them in viscous medium so that they can't reattach to the substrate, then over time they will undergo a process of terminal differentiation. Showing you this in more modern times, we've got some qPCR data down at the bottom, and you can see that cells at the starting point are enriched for cells expressing beta-1 integrins and other basal markers. This is the alpha-6 integrin, this is the transcription factor P63. But if you follow the cells over 12 hours in suspension, these markers go down. And in parallel, these markers, this protein involucrin that we work on a lot, and another marker, transglutaminase, go up. Now you'll see that it looks as though not much is happening between 0 and 4 hours when we look at any of these markers. But we found many years ago that evidence that at this point in suspension, 4 hours, although the cells still had integrins and were not differentiating overtly, they were committed to differentiate because they no longer responded to signals which would keep them in the stem cell compartment. And so in recent years, we've done a lot of work to try to understand what's special about these cells at the 4-hour time point compared to time 0. We've had some fantastic collaborations for this project. And essentially what we've done is to put the cells in suspension, isolate them, carry out in parallel transcriptomic analysis and proteomic analysis. And more recently, in work that we just published last year, we've also carried out lipidomic analysis. And we know from all of these studies that there's something distinct about this 4-hour commitment window. The aspects of commitment that we've pursued in most detail relate to protein dephosphorylation events that happen at 4 hours in suspension. We found when we looked at protein phosphatases in this assay, 22 protein phosphatases were upregulated at 4 hours. To find out if they had anything to do with exit from the stem cell compartment, we transiently knocked down expression of each of these phosphatases individually, and we carried out colony forming assays. So if you look at this spider's web diagram, each pale blue circle represents 10% of the cells forming colonies. The red circle is the scrambled control, so you can see a colony forming efficiency of just over 20%. And then if you look around the circle, you can see some examples of protein phosphatases like this one, DUSP6, where when you knock down expression, you increase colony formation. Now, what was interesting when we looked at the kinetics of the changes in the protein phosphatases with time in suspension, in contrast to basal markers, like P63, we could see that which go down with time, we could see transient peaks of expression of a number of the phosphatases as illustrated here. So our view was that upregulation of these protein phosphatases triggers cells to exit the stem cell compartment, and therefore they're acting to promote commitment. To summarize a large amount of work from many people in the lab, we realized that the protein phosphatases interact with one another positively and negatively at the RNA level over time. If we plot the level of 6 of these phosphatases and set it as 0 hours in the stem cell compartment, and then follow with time in suspension, you can see the RNA levels changing in this way. We realized that at the 4-hour time point, when many of them are upregulated, most of the interactions between the phosphatases were positive ones, which implied that this was in an unstable state. And we're really fortunate to be able to collaborate with a computational scientist, Sarah Jane Dunn, when she was at Microsoft in Cambridge. And she used Boolean network analysis to feed in our experimental data and show that it fitted a model where indeed the stem cell state is stable, the differentiated state is stable, and the phosphatases act as an unstable commitment switch. Now we know that we know quite a lot about how the protein phosphatases signal, and we know, for example, that they negatively regulate ERK-MAP kinase, which I told you is important to maintain stem cells downstream of integrins. And they also regulate AP1 transcription factors, which regulate genes like involucrin. So most recently, Toru Hiratsuka, who was until recently a postdoc in my lab, decided to look at the kinetics of ERK signaling, not on a population basis, but at the single cell level. And he did that using his expertise in FRET sensors. And he introduced into our cultured cells a FRET sensor for ERK-MAP kinase, which reports in the nucleus. And so this means that we could now look at the single cell level at changes in ERK dynamics. Another approach that Toru took was to introduce an M-Cherry fluorescent reporter under the control of the involucrin promoter to give us a fluorescent readout of differentiation in living cells. And what Toru found is illustrated in the 4 individual cells that you can see on the right-hand side here. So we're monitoring the FRET activity and involucrin levels over a period of about 12 to 16 hours. And in each of the 4 cells that you can see here, involucrin expression goes up because the red line has gone up. But we noticed that prior to the increase in involucrin expression, there was pulsatile ERK activity. And so this was really intriguing because it fitted with the idea that commitment is indeed an unstable state. So what we did next was to find out what effect our protein phosphatases had on ERK activity. So we had the ERK FRET sensor in our cultured human epidermal cells, and we introduced constructs in which we could overexpress protein phosphatases using doxycycline. And what we found for 2 of the protein phosphatases is illustrated here. So when we overexpress DUSP6, we see no effect on mean ERK activity, but we do see a significant reduction in pulsatile activity. Conversely, if we overexpress DUSP10, mean levels go down, but pulsatile levels are unchanged. And so the model that we have of what is going on as cells leave the stem cell compartment is illustrated schematically here. The stem cells have high stable ERK activity. The differentiated cells have low stable ERK activity. Pulsatile activity when mean levels are high means that it's not an irreversible indicator of commitment, because sometimes cells enter that pulsatile state, divide, and then return to the stem cell compartment. However, when we see pulses and mean ERK levels are lower, then that is indicative that the cells have become committed to differentiate and will downregulate ERK and switch on differentiation. So that is a summary of what we know from our in vitro experiments about the commitment state, how cells transition out of the stem cell compartment. And I should say that I didn't show it to you, but we've been able to do some very interesting in vivo imaging of the epidermis of mice that express the ferret sensor, and pulsatile ERK activity preceding differentiation is observed there too. Now, I told you that the stem cells are clustered rather than scattered in the basal layer of the epidermis. And I told you that we believe that delta signaling plays a role in controlling stem cell patterning. So I just want to show you what this looks like. Here we're labeling in brown with a particular stem cell marker called MCSP. And I'm showing you that in most body sites, the stem cell cluster lies closest to the surface of the epidermis, in contrast with these structures, which are called Rete ridges. Rete ridges are interesting and dynamic structures. Their height changes with age, and expansion and deepening of the Rete ridges is a hallmark of the inflammatory skin disease psoriasis. We were very anxious to find out more about the relationship between this undulation of the basal layer and the patterning of the stem cells. And we were able to look at that, first of all, by looking at, this slide shows you how the patterning of the stem cells looks when we look by whole mount labeling. So in these images here, we're looking at intact human epidermis, and we're looking at the basal layer by confocal microscopy. So here are the clusters of cells with beta-1 integrins where the stem cells lie. And when we look at the different protein phosphatases, we see that some of them, like DASP6, are co-expressed with this stem cells, but others, such as this one, PTPC7, have an inverse correlation. And some, like DASP10, show more uniform expression. So can we find out more about the importance of these topographical features? Well, to look at that, we used a bioengineering approach to make PDMS substrates in which the undulations, this is scanning EM of our substrates, but they mimic the dimensions of the undulations that you see in human skin. Now, we know if we plate keratinocytes on a flat surface, they will self-organize and give us integrin high clusters. But we found that if we plate the cells on a pattern substrate, the stem cells do indeed cluster, but they cluster in the, if you like, the right place. So here we're looking down on a region like this, labeling for the beta-1 integrins, and we can see indeed how they're clustered at the tips of the substrates. If we look at proliferation, we don't see patterning of that. But if we look at our differentiation marker involucrin, we can see that differentiation is, the differentiated cells are more abundant at the base of the surfaces. And this is quite intriguing, really, because the substrates are coated with collagen, but we're not applying ECM in a varied concentration. We also, if we look at the dynamics of ERK-MAP kinase signaling, can see that the cells with the highest ERK levels lie at the tips of these features where the stem cells lie. So the high level of integrin signaling is shown in red, and the cells with the lower levels are shown in green. So you can see that it's quite a dynamic substrate, but over the recording period, the cells with the highest ERK activity stay at the tips of the features. And then if we label for our different phosphatases, we find, sure enough, that DASP6 is most highly expressed at the tips of the features, whereas DASP10 shows a more uniform expression. So these artificial substrates are quite effective at mimicking the patterning of human epidermis. A couple of years back, we wanted to make a model system in which we could change the interface of the epidermis in culture from flat to undulating. And this is work that was led by Agilin Helling when she was in the lab. So we made this rig with a a membrane which could be deformed. We engineered a vacuum system, placed the membrane over some patterned holes so that when we applied the vacuum, the substrate deformed. This shows you again some scanning EM. It shows you some of the different sizes of the features that we could see. And it also showed how, according to, by applying different levels of vacuum power, we could achieve smaller or deeper undulations. Now, I'm not going to take you through the details of the next slide because the quantitation is quite complicated. But what we could show is that on our various different substrates, we induce patterning of cells so that the cells with the highest beta-1 integrins were in clusters, which differed from the cells with the highest integrin levels. But the bottom line from this experiment was actually that keratinocytes are rather dumb. And in this assay, they can't tell whether they're on the bottom of a feature or on the top. And this got us very interested in whether what is patterning the cells is not the substrates per se, but some of the physical forces that impact on the cells. Atifa Mabaseri in the lab, working with Sergi Garcia-Mañez, was able to test that by making AFM measurements of cell stiffness on the different substrates. So you can see the AFM probe. This one is on the tip of a feature and this one is on the base. This just shows you what the cells look like. And interestingly enough, we found that the stiffness of the cells on the base of the substrates was higher than the stiffness of cells on the tips of the substrate. And this differential cell stiffness could be abolished if we treated the cells with a Rho kinase inhibitor. So what we think is going on in terms of patterning of the stem cells in the basal layer is that the cells what they're reading is the physical forces which are exerted actually through cell-cell junctions and keep the stem cells in most body sites at the tips of the features. Now, we wanted to be able to look at cell-cell communication in more detail. And we were very fortunate to be able to come up with different approaches in which we could engineer cell-cell interactions in a more sophisticated way than we had previously. And one technique which we were able to develop, this is the work of Blaise Luis, was in collaboration with Zev Gartner and David Schaffner at UCSF. And this uses lipid-modified oligonucleotide to bring cells together. So the lipid enters the plasma membrane of the cells, and then complementary strands of DNA pair with one another to bring the cells together. And Blaise has shown very nicely that if we repeat our cell suspension experiment, but now use cells with the lipid oligos, we can induce clustering of the cells. What was somewhat disappointing, though, was that when we looked at whether that had an effect on differentiation, the answer was that it doesn't. So if we look at the kinetics of induction of involucrin, for example, over time, there's no difference between the single cells and the clusters. We've done a lot of analysis of the different protein phosphatases, and though in this particular experiment, it looks as though there is a difference between single cells and clusters. Actually, many experiments have shown that intercellular adhesion is not changing the induction of the protein phosphatases or altering whether or not the cells differentiate. So that brings me really back to the Notch ligand delta-like 1, because the concept that cell-cell adhesion is not important in regulating differentiation doesn't sit very well with our previous findings. And what I've shown in this slide is some more recent experiments in which we have plated single cells on extracellular matrix, either alone or in the presence of the Notch ligand jagged 1, which we know induces differentiation. And then we've looked at what are the effects of overexpressing delta. So I'll just draw your attention to this experiment here. These are cells expressing the empty vector, And we can show that if we expose them to jagged 1, we stimulate induction, we induce differentiation. But if the cells are also expressing delta-like 1, that induction of differentiation is obviated. So delta expression by the stem cells is not only stimulating neighboring cells to differentiate, but under certain circumstances, it can inhibit differentiation cell autonomously. Now, one problem that we've always had with delta is that it is expressed in very low levels in human epidermis. Sometimes in despair, we describe it as being expressed at homeopathic quantities, and it's really difficult to understand why it might be so important. But recent advances came through the work of Victor Negri, who, through knowledge of Notch signaling, realized that key proteins which affect the Notch pathway are known as fringe proteins. And these are N-acetylglycosyl transferases, which modify or add sugars to Notch EGF repeats in the Golgi complex. And there are 3 of these in mammalian cells. They go by the name of lunatic fringe, manic fringe, and radical fringe. And what makes them particularly interesting is that lunatic and manic greatly increase the affinity of Notch for delta-like 1, and they can inhibit the receipt of jagged receiving signals. So when we had a look in our cultured keratinocytes, we found that lunatic fringe was by far the most abundant fringe protein. So we set out to find out whether lunatic fringe expression could modulate the properties of keratinocytes. And without taking you through all of the panels of this slide, you can see over here that if we overexpress lunatic fringe in our cultured keratinocytes, we reduce the expression of our differentiation markers. And in our colony forming assays, you can see that we massively increase colony formation. And so we have a concept now where delta acting in concert with lunatic fringe is playing a role in the stem cell compartment, clustering the stem cells, stimulating neighboring cells to differentiate, and protecting the stem cells from differentiating themselves. So everything that I've told you so far is based on experimental manipulation of cultured human epidermal cells. And we all love our experimental models, but at some point we need to look up from the bench and ask, what is the relationship of what we're seeing in vitro to the in vivo situation? And so I'm just going to end with some observations from this initiative, which is called the Human Cell Atlas. It was launched in 2016 by Aviv Regev and Sarah Teichmann. And the goal is to map all of the cells in the human body, to use that information to get new insights into normal physiology and disease mechanisms, and also to drive new approaches to data integration, collaboration, and data sharing. I was very excited to be part of this because I, of course, wanted to test the predictions from my studies on cultured human epidermis. So is it true that there are multiple populations of cells in the basal layer? Is it true that there's a transient upregulation of protein phosphatases associated with commitment? And is it true that there's differential expression of Notch pathway components in different regions of the epidermis? Well, we've been able to answer those questions and many more through a fantastic collaboration led by Sarah and Muzz, and at the KCL end, led by Magnus. And so what we have now is access to single-cell RNA sequencing for over 500,000 cells, not just from the epidermis, but also from the dermis, and also from immune cells and blood vessel cells. We've got data from healthy adult skin, skin from patients with atopic dermatitis and psoriasis, and there are also some samples of developing skin from fetal tissue. So if we take this huge data set and simply focus in on the epidermis of healthy patients, what do we find? And this is the analysis which has been carried out by Victor Negri. a wonderful thing about the epidermis is that we know so much about the markers of the different layers that it's very easy to say immediately these populations of cells in the UMAP plots belong in the basal layer, these ones belong in the differentiating cell layers. But if we start to look in more detail here, you can see that the plot maps 3 distinct populations of basal cells. So what are they? We could barely contain ourselves when it came to looking at the data. So these are UMAT plots from individual candidate markers. And here you can see some of the markers I've told you about. So we see high expression of beta-1 integrins in this population called basal 3. Interestingly enough, high expression of delta-like 1, although it's expressed at low levels, in basal 2, together with the transcription factor p63. And then 2 other markers that we had found, caveolin 1 and 2, are enriched in basal 1. Now, to some extent, this is a puzzling finding because by flow cytometry, the beta-1 integrins and CD46 are co-expressed in the same populations of cells. So we're now working with our collaborator in Dundee, Angus Lamond, to find out whether, although these populations differ at the transcript level, they, in terms of the protein level, they are indeed similar. For example, we know the half-life of the beta-1 integrins is considerably shorter than the half-life of caveolin 1 and 2. Now, if we go back to the protein phosphatases, the question is, can we pick up a population of cells that correspond to that transit from the basal to the suprabasal compartments? And sure enough, when we look in the data sets, we pick out these 2 populations here, which we call transition 1 and 2. And if we look at our protein phosphatases, they map very nicely there. And in particular, if we go back to the qPCR data I showed you earlier, we always knew that the peak of DUSP6 expression was much tighter than the peak of DUSP10 expression. And you can see that that really fits extremely nicely with the UMAP plots of cells from human epidermis. Now, last but by no means least, what is going on with the Notch pathway? Well, we were able to confirm what we knew about expression of DLL1 in the basal 2 population. You can see also, though, that this is where jagged 1 and 2 are most highly expressed. And so that's why the protective effect of DLL1 is important. We know as cells differentiate, Notch signaling is upregulated, and you can see evidence for that when we look at some of the markers of the Notch pathway. And again, these are shown down here. And last but by no means least, what about the fringe genes? This is the data I showed you from the cultured epidermis, and we're really pleased to see, as predicted, that lunatic fringe is most highly expressed of the 3 fringe genes in the UMAP plot. And sure enough, it is at the RNA level most highly expressed in the basal 2 population, along with DLL1. So the last things that I want to tell you about really are where we're going next. Using all of the data that we have from the Human Cell Atlas, we can compare the relative size of different populations of epidermal cells in healthy and diseased skin. And this is important because it allows us to generate more hypotheses that we are able to test experimentally. And the other thing that we're really, really keen to do is to be able to diagnose accurately the composition of different cells in the skin simply by running an OCT probe over the skin surface. So what you can see here is running the probe over the skin, building up a 3D slice. You can see when this tips as it comes round. This is the surface of the epidermis. And then as we slice through that, we can easily distinguish the boundary between the epidermis, the dermis, different layers of the dermis, and different blood vessels. And we can use software to give us a very nice picture, for example, of how the vasculature looks in different body sites. So our vision now is to be able to integrate across the different scales the information that we have on the different cell types in the skin. So to conclude, single-cell RNA sequencing as part of the Human Cell Atlas has confirmed our predictions about multiple populations of cells in the epidermis and dermis. It's been a reality check for our experimental models. It's given us new ways to try to integrate different data sets. And it's giving us some really interesting approaches to developing new hypotheses to test. And then the last slide is the most enormous thank you to my lab. This was Christmas 2019. Since then, we've been nowhere near so close together, at least physically, but we've remained incredibly close as a team. And I'd like to pay tribute to all of them for just being fantastic colleagues. So with that, I'll stop and congratulate the London Metallomics facility on a great launch. Thank you.